for all that stuff. Cytoplasm. Cytoplasm is everything between the cell membrane and the nuclear membrane. Cytoplasm is everything between the cell membrane and the nuclear membrane. The liquid part is called cytosol. The liquid part is called cytosol. Floating around in that cytosol are organelles. Floating around in that cytosol are organelles. The word organelle means little organs. They're not technically organs, but they got jobs like organs. Cytosol plus organelles is cytoplasm. Clear? All right, now take a look at page 83. First one we're going to talk about are mitochondria. From lab, give me a function for mitochondria. Produce ATP, beautiful, and never say the powerhouse of the cell, don't do it. All right? They produce ATP. They're really cool little things, they look like little worms. And one of the things they divide, like bacteria. One of the things that's fascinating about mitochondria is they have different RNA than us. They have different DNA than us. They have bacterial DNA. We have fancy chromosomes. Bacteria have a single circle of DNA. Bacteria have different ribosomes too. Mitochondria have the same ribosomes as bacteria. They have the same DNA as bacteria. We're pretty sure why too. Long time ago, mitochondria were free-living bacteria and took up residence inside other cells. They found themselves a nice house. That cell fed them, protected them, and in return, they gave it ATP or energy. That's what let single-celled creatures become multi-celled creatures, become us. So mitochondria are actually leftovers of our ancestors, our single-cell ancestors. They're actually little bacteria living inside the cell. Now they've since evolved and they're not going to live by themselves anymore. But that's where they come from. And if you look on page 83, you see they have folds inside of them. Everybody see those folds? It's like kind of like a folded up blanket inside the mitochondria. Those are called cristae. Those are called cristae. They form different chambers in the mitochondria. They form different chambers in the mitochondria, and that's where ATP synthesis is actually taking place. That's where the ATP synthesis is actually taking place. The mitochondria produce ATP, and they do it on cristae. Next up, ribosomes. What do ribosomes do? Make proteins. Good. Ribosomes make protein. They're actually kind of like little machines that assemble proteins. You can see a picture of a ribosome on page 94. But if you want to see a bigger picture of a ribosome, take a look at page 105. If you want to see a really big picture of a ribosome, page 106, 107. Ribosomes are actually the machines that assemble proteins. Ribosomes are the machines that assemble proteins. They come in two parts. You don't need to worry about the two parts, but you can see them really well on 106, 107. You'll find ribosomes two different places. You can either find them as free ribosomes or membrane-bound ribosomes. Free ribosomes or membrane-bound ribosomes. They are the same organelle. A ribosome can be floating through the cytosol. They call it a free ribosome. Then it can sit down on the endoplasmic reticulum. And we call it a membrane-bound ribosome. It's the same ribosome, and it can be one day and the other one the next day. It's just where it is. That's how we name it. Does that make sense to you guys? Free ribosomes are floating free in the cytosol. Membrane-bound ribosomes are stuck to membrane. Both of them make protein. The membrane that they stick to can be endoplasmic reticulum. Take a look back at page 105. There are some ribosomes sitting on rough endoplasmic reticulum. There are some ribosomes sitting on rough endoplasmic reticulum, page 105. They're all kind of lined up. And they're actually kind of pooping their proteins down into the rough ER. That's why we say rough endoplasmic reticulum is a site for protein synthesis. Because you have a whole bunch of ribosomes stuck to it, squirting proteins down into the membrane. Kind of collects them. I'm going to show you guys a video in a little bit. Make this a little clearer. So, rough endoplasmic reticulum is a site for protein synthesis. Ribosomes are making the proteins. And when they're stuck to the rough ER, we call them membrane-bound ribosomes. Does that make sense to everybody? You guys are all right? Okay. Golgi apparatus. You will also see it called Golgi body. I don't care which one you use. You should recognize both of them. If we go back to page 86, you can see it on page 85, but 86 kind of puts everything together for you. The Golgi apparatus, or Golgi bodies, or Golgi complex, packages, modifies, and transports cellular products. Packages, modifies, and transports cellular products. What happens in the cell is the ribosomes make the proteins into the rough ER. The rough ER kind of bags it up into a vesicule. Remember, a vesicule is a membrane-bound bag of stuff. That vesicule full of proteins is carried from the rough ER to the Golgi apparatus. It's put into the Golgi apparatus where it's changed. There are enzymes in there that will change the protein into its final form. And then the Golgi apparatus will package all those changed proteins together and ship it where it needs to go. It may need to go somewhere inside the cell. It may need to go outside of the cell. If it has to go outside of the cell, what's the name of the process the cell's going to use to kick it out? Exocytosis. Is exocytosis an active process or a passive process? Active. It takes ATP, right? How do we feel about that, okay? You guys pretty much understood everything I said there? If you didn't, ask questions. All right. The ribosome produces a protein, kind of squirts it into the rough endoplasmic reticulum. Rough endoplasmic reticulum bags it up into a vesicule. That vesicule is carried to the Golgi apparatus. The Golgi apparatus takes it in one side, changes it chemically, packages it up, shoots it out the other side. Now it's ready for export. There it can be taken somewhere in the cell if it's needed, like maybe it's a lysosome, or it can be taken to the plasma membrane and released outside through exocytosis. Okay? lysosomes, what did I say they were in lab? Little stomachs, yeah, lysosomes are like little stomachs. They're little bags of acid. Page 86, you can see a little paragraph on lysosomes. Lysosomes are membrane-bound vesicles, bags of stuff, and that stuff is actually called acid hydrolases. That stuff is called acid hydrolase. There's different ones. Hydrolase is the word that ends in ASE. What does that tell us? It's an enzyme that breaks stuff down. It's an enzyme that breaks stuff down. This is like acidic digestive juice inside the lysosome. This is acidic digestive juice inside the lysosome, like a little stomach. So the cell uses lysosomes to do a couple things. It helps it break down worn out organelles. Lysosomes help the cell break down worn out organelles. Those worn out organelles are recycled. Lysosomes can also help digest any particle taken inside by endocytosis. Lysosomes can also help digest any particle taken inside by endocytosis. Certain cells will actually eat bacteria. They'll do phagocytosis and go and eat a bacteria. Then that cell has that bacteria in a little bag, a vesicle, inside. And I'll take a lysosome and connect that lysosome to the bag with the bacteria, and that dissolves the bacteria. Bacteria is killed, it's dissolved by the acid hydrolase in the lysosome. That make sense? All right. Peroxisomes are also membrane-bound vesicles. Peroxisomes are also membrane-bound vesicles. They contain chemicals called oxidases. They contain chemicals called oxidases. Oxidases help detoxify harmful chemicals. Oxidases help detoxify harmful chemicals. They use oxygen to do it. And again, it's an A, it's ASE, it breaks down. It uses oxygen to break down harmful chemicals, like alcohol, the drinking kind, ethyl alcohol. Peroxisomes are particularly prominent in cells of the liver. Lots of peroxisomes in liver cells, because that's the organ that breaks down the alcohol. Now take a look at page 88. The cytoskeleton actually means cell skeleton. It's a bunch of tubes and fibers inside the cell. Cytoskeleton is a bunch of tubes and fibers inside the cell, and there's 3 different kinds of fibers. Page 88, you can see them: microfilaments, intermediate filaments, and microtubules. On page 88. Microfilaments, intermediate filaments, and microtubules. Microfilaments are the smallest, the thinnest parts of the cytoskeleton. They help the cell move around and change shape. They help the cell move around and change shape. These are also found in muscle cells. They help the muscle cells contract. They help the muscle cells contract. Without microfilaments, we couldn't move. They also form the cleavage furrow. You guys remember cleavage furrow from lab? These guys are the ones that make the cleavage furrow, so you can have, what's the name of that process? Cytokinesis. Remember, cytokinesis is dividing cytoplasm. Mitosis is dividing nuclei. Huge difference. They get lumped together. Huge difference. Intermediate filaments are kind of in between. Intermediate filaments are in between. Intermediate filaments are found in desmosomes. Intermediate filaments are found in desmosomes. They actually attach to them and help the cell avoid being pulled apart, help the cell resist tension. Make sure you can't just split the cell open. You guys remember desmosomes? What are they? Anchor points? Yes, anchor points are cell junctions between cells, places where cells are attached that are really strong. Intermediate filaments run from the desmosomes into the cell, so the cell doesn't get pulled apart. And then there are the microtubules. Microtubules are the biggest of those three. Microtubules are the biggest of those three. Microtubules form the mitotic spindle. You knew that already. Microtubules form the mitotic spindle. They make up the mitotic spindle. They are attachment points for organelles. They are attachment points for organelles. And there are highways for little dudes that walk around inside your cell. There are highways for little dudes that walk around inside your cell. I'm not kidding. They're not actually little dudes, but they look like it. They're called motor proteins. They walk along the microtubules carrying stuff. They're pretty cool. They're going to be on the video that I show you. The motor proteins are the walking microtubules of the tube they walk along. That's the cytoskeleton. The centrosome is the area where you find centrioles. The centrosome is the area where you'll find a centriole. Not so worried about centrosomes. Centrioles are important. From lab, what do centrioles do? Yeah, they form the mitotic spindle. They make microtubules. Centrioles make microtubules, form the mitotic spindle, and they direct chromosome division during mitosis. They actually move the chromosomes around and split them during mitosis. They move the chromosomes around and split them during mitosis. Page 89, you can see a centriole there. They're like the L-shaped little tubes. And then page 90 and 91. Cilia and flagella. They kind of look alike, but they're not the same thing. Cilia are little hair-like extensions from cells. Cilia are little hair-like extensions from cells. And they wave back and forth like this. Sit there and they wave back and forth like this. Their function is to move material. Their function is to move material, very often mucus. move material, very often mucus. They are what brings that dirty mucus up out of your lungs. You're breathing in dirt and dust every day. Gets caught in the mucus on your bronchi and your trachea, brought up here, and dumped down your esophagus. If you have meat for life, you know how much snot you eat a day. How much? A quart. You eat a snot a quart a day. You eat a quart of snot a day. And it's dirty. But it gets dumped in your stomach, which is full of snot anyway. So no big deal. Unless what? What makes your cilia not work? You're a smoker. You smoke. You paralyze your cilia. And all that junk stays down in there. It doesn't come out. Anybody a smoker in here? Everybody wants to admit it? That's cool. Don't start. I've quit. Again. This one's sticking for a while now. I'm doing good. First thing you do when you're a smoker, you wake up in the morning, you got a rattly cough. There's snot moving around in there. So what do? You fire down another cigarette, paralyze everything so it doesn't rattle around so much. Keep all that junk down in there. Don't start. Cigarette smoke kills cilia. They grow back. Body's remarkably tolerant if you grow back. Flagella have a different function. Cilia move material. Flagella, they cells swim. What am I talking about? Sperm cells. That tail on a sperm is called a flagellum. So flagella move the cell. Cilia moves other stuff. Cilia moves other stuff. There are inclusions, which are storage areas for your body. If an inclusion has glycogen in it, we call it a glycosome. It's got a fancy name. An inclusion filled with glycogen is called a glycosome. Now, what is glycogen? You should know this right off the top of your head. What's glycogen? Storage form of glucose. Yes, good. That's the way your body stores glucose. It chains it all together to make glycogen. Chains it all together to make glycogen. If you didn't remember that, you better go back and study organic molecules for the test. Promise. Some cells have little inclusions with lipid in it. Those are fat cells. Fat cell looks kind of like this. We'll look at a more living tissue. Anybody know the fancy name for fat cell? Adipocyte, yes, good. That's an adipocyte. All this stuff in here is liquid fat, lipid. Basically a big balloon filled with lipid. That's an adipocyte. Sometimes you'll find melanin in inclusions. Where do you find cells that have melanin? In your skin. Melanin is a pigment. Melanin is a pigment. What color is it? Brown. Yeah, it's brown. Melanin is brown. The more melanin you have, the more brown you are. Why do we have melanin? What's its function?
Protects UV light.
Protect you from UV rays, yes. Melanin protects you and your skin from UV rays. That's why if you have fair skin, if you have lighter skin, you go out in the sun, what happens to you?
You get
You
burned.
burn. You do it over and over and over again, what happens to you?
You get
You tan eventually, yeah, you get melanoma. You tan, some people don't tan, some people do tan, other people have nice brown skin anyway, they don't need to worry about that, they still tan too. But, melanin is protective. One of the reasons why we have so many different skin colors is because our ancestors come from different parts of the planet. If your ancestors came from an area near the equator, you have more melanin. If your ancestors come from far away from the equator, you have less melanin. Because you need to get vitamin D from the sun. Sun converts vitamin D in your skin. So if you have too much melanin and you don't get a lot of sun, you don't have enough vitamin D. If you get too much sun, you get skin cancer. So it's kind of like a balance. We're all different colors because of that. So if you have more melanin, do you have more melanocytes? Melanocytes are the cells that make melanin. So if your skin's darker, you have more melanocytes? No, everybody's got the same number of melanocytes. If your skin's darker, your melanocytes are better, they're harder working. You got really white skin, you got some lazy melanocytes. You got brown skin, you got melanocytes that are working. All right, nucleus. Take a look at page 92, there's a nucleus. The nucleus is separated from the rest of the cell by the nuclear membrane. Nucleus is separated from the rest of the cell by the nuclear membrane. Or nuclear envelope, same thing. Nuclear membrane, nuclear envelope, same thing. That is a double phospholipid bilayer. There's two phospholipid bilayers there. The nuclear envelope has pores in it to let RNA from the nucleus out into the cytoplasm. Just write that down, it won't make sense yet, but just write it down. The nuclear envelope has pores in it to let RNA move from the nucleus out into the cytoplasm. Nucleoplasm is a fancy word for the cytoplasm inside the nucleus. Fancy word for cytoplasm inside the nucleus, nucleoplasm. Bless you. Nucleoli are the little ball-like structures inside the nucleus. You can see one on page 92. You can see one on page 92. The cell models in the lab have two of them. Nucleolus is singular. Nucleoli is plural. Nucleoli make ribosomes. Nucleoli make ribosomes. And then on page 93, you can see chromatin. Chromatin is normally how your cell keeps DNA. It's the normal form of DNA. Chromatin is the normal form of DNA. You guys already know DNA is like the double helix. Chromatin is when you take that double helix and wrap it around proteins called histones. Histones are the proteins. They're kind of like coat hangers for the DNA. Wrap your DNA around the histone, and that makes a long string called chromatin. Chromatin is the usable form of DNA. Chromatin is the usable form of DNA. You can get to it. Your cell can use it as a recipe book for proteins, right? Because that's what DNA is, recipe book for proteins. Now, chromatin, you can get to the DNA, you can read it. when the cell gets ready to divide, that chromatin condenses into chromosomes. Look on page 93, the bottom picture there. There's a little X. When you hear chromosome, that's what you think, like a little X-shaped thing. That is the moving form, the packed up form of DNA. You can't use it, but it's going to be easy to divide equally. It's going to be easy to divide equally. You guys been working hard for the last half hour. Let's watch a little video here. Any questions? Any questions about where we are, what we're talking about? Everybody okay? A histone is a protein that helps hold DNA. Kind of like keeps it organized. Think about a coat hanger. And take your jacket on, put on a coat hanger and hang it up so it doesn't get tangled up. Histones are holders for DNA when it's in the form of chromosomes.
What's a glycogen build inclusion?
Glycosome. Glycosome. All right, what I'm going to show you is kind of like an animation of the artist concept of what's going on inside the cell. But I don't want you to think this is just kind of some randomly drawn thing. Ooh, I know what Snoopy looks like, so here's Snoopy. I know what Mickey Mouse looks like, here's Mickey Mouse. If you still got your books open at page 92, there are some electron microscope pictures of the nuclear pore complex, of the nucleus itself. You can see the inner and the outer membrane, and also some of the proteins that make up the nucleus. We have microscopes that can see that stuff. We have microscopes that know what this stuff looks like, so it's not just Joe Animator going, ooh, I think I'll draw Patrick and SpongeBob. This is what it looks like. This first one doesn't have any animation. I just want you to watch this. See what's going on, and see if you can recognize any organelles, recognize any cellular processes, recognize where you are. And enjoy the music, it's kind of fun. There is a narrated version of this, lots of big words. I wanna talk about it first, and I'm gonna show you the big words one. The reason I'm showing you this thing like 3 times is because I think it's really important, and I think it's gonna help you put some of this stuff together. So bear with me. Don't be going like, oh my God, I'm so bored just yet. Alright? Where are we? Yeah, we're inside a blood vessel. This is the walls of the blood vessel. These things are red blood cells. These things are white blood cells. Watch them fly by. See how the red blood cells are whipping past really fast? And the white blood cells are just kind of rolling along like tumbleweeds. The reason they're not whipping by really fast is because of this. See, this is the white blood cell, and this is the wall of the blood vessel. The wall of the blood vessel is made of a tissue you learned in lab. Take a wild guess at what tissue might line the inside of blood vessels. So close. Yeah, simple squamous epithelium. These are tubes made of simple squamous epithelium. So the white blood cell here and the simple squamous epithelial cells here are interacting with each other in this fuzzy area. Watch what the fuzzies do. See, the fuzzies are kind of grabbing each other as it rolls past, so it's kind of like Velcro. Not as quite sticky. Velcro is what sticks your epithelial tissues on the connective tissue. Remember basement membrane? Not quite that sticky, but it's sticky enough that it's slowing the white blood cells down while it's rolling along. How we doing? Is this making sense? All right. This is the plasma membrane. Phospholipid bilayer. Watch these balls kind of float around. It's like ping pong balls in a bathtub. Did you actually try that? You don't have enough ping pong balls, right? I get it. Remember we talked about lock and key? This molecule from, let's see, this is going to be from the endothelial cell, I think. And this is going to be from the white blood cell. Maybe vice versa. Gotta watch this, figure it out. They fit together though. See how this piece fits in here? This sends a signal inside this cell. This is going to signal this cell, hey, do something. Because this key fits in this lock. That's something fitting in a receptor. This would be a receptor. It receives a message and sends a message to this cell. Now we're kind of falling back into the cell, looking up at the cell membrane. This is the cell membrane, and outside the cell, we're falling down into the center of the cell. This stuff is active. See, they were forming an actin microfilament. The reinforcements here. Now we're going to tear one apart. Cells build this stuff and tear it down all the time. This is a microtubule. This looks bigger than the microfilament, doesn't it? This is a big old tube built of these things called subunits. Now we're tearing one apart. This is the walking dude, motor protein. Each time it takes a step, it's burning an ATP. Like step, 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 ATP, ATP, ATP. It's carrying a vesicule. Vesicles are bags of stuff. This is a membrane-bound bag of something. We don't know what it is. But it's a bag of stuff, and this guy's carrying it to where it needs to go. One in the background, too, carrying his bag. Alright, there's several things here you should be able to see. What's this big old goofy thing here? That's a mitochondria. What's that? Centriole. What's this big giant thing? Nucleus. And what's this cloudy looking stuff? Rough endoplasmic reticulum. Yeah, keep an eye on it as we go along. There's the centriole. This is the surface of the nucleus. These are pores. The stuff coming out is RNA. And what RNA is? RNA is a recipe for protein. There's a loop of RNA. If you've seen this stuff before, this is messenger RNA. We'll get to that. Hopefully today, maybe tomorrow, maybe next class. There's a ribosome forming. There he is. Ribosome is the machine that makes proteins. It's going to read messenger RNA. Here's the messenger RNA. That messenger RNA is going to code for amino acids. The ribosome here is putting them together one at a time, one at a time, one at a time. And here's the protein. That is a message, maybe for this mitochondria, a chemical message, maybe for that mitochondria. Here we have another ribosome. It's stuck to a membrane. So what is this thing right here? Rough endoplasmic reticulum. That ribosome stuck to it. It's going to poop its little protein out into the rough ER. Now it's time to be free. Now these vesicles are headed towards the Golgi apparatus, being carried by the walking dude. Walking dudes are carrying all of these. Golgi apparatus. Exocytosis. This cell is releasing this chemical message to, pretty sure this is the white blood cell. Because if this is an endothelial cell that's signaling, hey, there's an infection here, I'm going to release some chemicals to tell the white blood cell, we need help, we need help. See how this got real solid? That's called a raft. There's more cholesterol in there. It's more solid than the rest of the stuff around it. And all these proteins are going to communicate with this cell. So the white blood cell says, hey, wait a minute. I'm getting a message, I need help. So it's going to flatten out and squeeze out between this cell and this cell. The squamous epithelial cells. It's going to squeeze out of the blood vessel into the tissue around it. So what do you guys think of that? Kind of interesting, isn't it?
Can't believe how complex it is.
Oh yeah, this is kind of just touching on it.
Billion years of evolution.
Yeah, it's amazing stuff. If you want to see this, you want to show it to your friends?
Yeah.
Google Harvard Cell Bio video or Harvard Cell Biology video. If they ask you, hey, how's anatomy class going? Go, you should check this out, cause I know what's happening here. Let's see, it's 8:57. You guys wanna see the full narrated 8-minute video or not?
Yeah.
You promise you're gonna stay awake and not text?
Yes.
I see a couple of you. Just saying.
While red blood cells are carried away at high velocity by a strong blood flow, leukocytes roll slowly on endothelial cells. E-selectins on endothelial cells interact with ESGL1, a glycoprotein on leukocyte microvillum. Leukocytes, pushed by the blood flow, adhere and roll on.
Leukocyte means white blood cell. What does erythrocyte mean?
Red blood cell.
Red blood cell.
Endothelial cells because existing interactions are broken while new ones are formed. These interactions are possible because the extended extracellular domains of both proteins emerge from the extracellular matrix, which covers the surface of both cell types. The outer leaflet of the lipid bilayer is enriched in sphingolipids and phosphatidylcholine. Sphingolipid-rich rafts raised above the rest of the leaflet recruit specific membrane proteins. Rafts' rigidity is caused by the tight packing of cholesterol molecules against the straight sphingolipids hydro.
There's a nice cutaway view of the inside of the phospholipid bilayer. And see how this kind of glue like this? Ping pong balls on the back.
Carbon chains. Outside the rafts, peaks in unsaturated hydrocarbon chains and lower cholesterol concentration result in increased fluidity. At sites of inflammation, secreted chemokines bound to heparin sulfate proteoglycan on endothelial cells are presented to leukocyte.
Okay, this is the endothelial cell. This is the leukocyte. This is the receptor.
Seven transmembrane receptors. The binding stimulates leukocytes and triggers an intracellular cascade of signaling reactions. The inner leaflet of the bilayer is a very different composition than that of the outer leaflet. While some proteins traverse the membrane,
What would you call this category of protein?
Column.
Transmembrane protein. Goes across the whole membrane, right?
theirs are either anchored into the inner leaflet by covalently
Peripheral protein.
attached fatty acid chains, or are recruited through non-covalent interactions with membrane proteins. The membrane-bound protein complexes are critical for the transmission of signals across the plasma membrane. Beneath the lipid bilayer, spectrin tetramers arranged into a hexagonal network are anchored by membrane proteins. This network forms the membrane skeleton and contributes to membrane stability and membrane protein distribution. The cytoskeleton is comprised of networks of filamentous proteins that are responsible for the spatial organization of cytosolic components. Inside microvilli, actin filaments form tight parallel bundles, which are stabilized by cross-linking proteins. While deeper in the cytosol, the actin network adopts a gel-like structure, stabilized by a variety of actin-binding proteins. Filaments, kept at their minus ends by a protein complex, grow away from the plasma membrane by the addition of actin monomers to their plus end. The actin network is a very dynamic structure, with continuous directional polymerization and disassembly. Severing proteins induce kinks in the filament and lead to the formation of short fragments that rapidly depolymerize or give rise to new filaments. The cytoskeleton includes a network of microtubules created by the lateral association of protofilaments, formed by the polymerization of tubulin dimers. While the plus ends of some microtubules extend toward the plasma membrane, proteins stabilize the curved conformation of protofilaments from other microtubules, causing a rapid plus end depolymerization. Microtubules provide tracks along which membrane-bound vesicles travel to and from the plasma membrane. movement of these cargo vesicles is due to a family of motor proteins linking vesicles to microtubules.
These guys are actually very close related to the myosin protein that makes your muscles contract. You're just using the same thing in two different places for two different tools.
Membrane-bound organelles like mitochondria are loosely trapped by the cytoskeleton. Mitochondria change shape continuously, and their orientation is permanent,
Just like a little bacteria swimming around in the soup.
dictated by their interaction with microtubules. All the microtubules originate from the centrosome, a discrete fiber structure containing two orthogonal centrioles and located near the cell nucleus. Pores in the nuclear envelope allow the import particles containing mRNA and proteins into the cytosol. Here, free ribosomes translate the mRNA molecules into proteins. Some of these proteins will reside in the cytosol. Others will associate with specialized cytosolic proteins and be imported into mitochondria or other organelles. The synthesis of cell-secreted and integral membrane proteins is initiated by free ribosomes, which then dock to protein translocators at the surface of the endoplasmic reticulum.
There we got the ribosome stuck to this port or passageway that produces the protein inside our endoplasmic reticulum.
Nascent proteins pass through an aqueous pore in the translocation. Cell-secreted proteins accumulate in the lumen of the endoplasmic reticulum, while integral membrane proteins become.
This is how you make the proteins, like the proteins that are in the plasma membrane, already in the membrane. This is already in there. And watch what happens, how they get it up to the surface.
Embedded in the endoplasmic reticulum membrane. Proteins are transported from the endoplasmic reticulum to the Golgi apparatus by vesicles traveling along the microtubules.
See those proteins on there?
Protein glycosylation, initiated in the endoplasmic reticulum, is completed inside the lumen of the Golgi apparatus. Fully
See, there's that same bag being carried up towards the surface. And look, it's studded with those proteins that the ribosomes made and left in the membrane.
glycosylated proteins are transported from the Golgi apparatus to the plasma membrane. When a vesicle fuses with the plasma membrane,
See, there's the proteins that were in the bag. And here are the proteins that were part of the bag.
proteins contained in the vesicle's lumen are secreted, and proteins embedded in the vesicle's membrane diffuse in the cell membrane. In sense of inflammation, chemokines secreted by endothelial cells bind to the extracellular domains of G protein-coupled membrane receptors. This binding causes a conformational change in the cytosolic portion of the receptor, and the consequent activation of a subunit of the G protein. The activation of the G protein subunit triggers a cascade of protein activation, which in turn leads to the activation and clustering of integrins inside lipid rafts. A dramatic conformational change occurs in the extracellular domain of the activated integrins. This now allows for their interaction with ICAM proteins displayed at the surface of the endothelial cells. These strong interactions immobilize the rolling leukocyte at the site of inflammation. Additional signaling events cause a profound reorganization of the cytoskeleton, resulting in the spreading of one edge of the leukocyte. The leading edge of the leukocyte inserts itself between endothelial cells, and the leukocyte migrates through the blood vessel wall into the inflamed tissue.
So there you have it. That's just one tiny process, what's going on in your body all the time. That's talking to each other with chemicals, doing all kinds of crazy stuff. So again, if you wanna watch that a couple times, show it to your friends, they'll all go, ooh, you're learning all this crazy stuff, it's kinda fun. How are we doing, any questions? Alright, we got like 7 minutes left, so let's knock this stuff out. Cell life cycle video. You all saw this in lab, right? Is there anybody who has not talked about the life cycle of the cell in lab? Raise your hand. Okay, you're gonna be, you're Friday, right? Okay. I'm trying to think. Did you talk about mitosis at all? Okay, this is kind of that. If you have me for lab, you've seen this video already, right? Prophase, metaphase, anaphase, telophase. You go to prophase, metaphase, anaphase, telophase? Okay. I'm gonna try new video. Have not seen this one. This one I use all the time. It looks like it came off a film strip. You guys know what film strips are? Way back in the dark ages, like when I was in school, they used it as a little projector, and it had like a piece of film, and you'd play a cassette tape, and it would go beep, and you'd click to the next picture and play blah, 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 beep, and you'd click to the next thing. Really old school. This looks like it came off of there. This one is all fancy and new and look 3D. Let's watch this just for fun.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day, our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into 2 identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or Gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or Gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as 2 sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together. freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot...
Okay. Those of you who saw both, how many would like this one better? How many would like the other one better, the old school one? Okay, so we got kind of a mix. I don't really have time to show you this one today. Tell you what, next class we'll go over this a little more. We'll talk more about interphase and mitosis, and then move on to the rest. If you have questions, if you're not sure, think about questions you might have for the beginning of the class. So if you have any questions or anything, keep them in mind. You may have a quiz on Monday, it's certainly fair game. Everybody okay? Questions, comments, concerns? Fly, be free. Now you can go.